Pacific Biosciences is developing a transformative DNA sequencing technology that will revolutionize the field of genetic analysis by enabling researchers to answer questions important to human healthcare. We call it SMART, Single Molecule Real-Time DNA Sequencing, a breakthrough technology based on the natural process that occurs every time living cells divide. Prior to division, DNA is replicated by enzymes called DNA polymerases, which efficiently duplicate entire genomes in minutes by reading the DNA and sequentially building a complementary strand with matching building blocks called nucleotides. Pacific Biosciences' SMART sequencing harnesses the power of the polymerase as a sequencing engine by eavesdropping on it while it works to replicate DNA. This approach is enabled by two proprietary technologies. The first is phospho-linked nucleotides. To visualize polymerase activity, a different colored fluorescent label is attached to each of the four nucleotides, A, C, G, and T. In contrast to other sequencing approaches, our phospho-linked nucleotides carry their fluorescent label on the terminal phosphate rather than the base. Through this innovation, the enzyme cleaves away the fluorescent label as part of the incorporation process, leaving behind a completely natural strand of DNA. This enables us to exploit the inherent properties of the DNA polymerase, including high speed, long read length, and high fidelity. The second key technology is a nanophotonic visualization chamber called the Zero Mode Waveguide, or ZMW. It enables observation of individual molecules against the required background of labeled nucleotides, while maintaining high signal-to-noise. The ZMW is a cylindrical metallic chamber approximately 70 nanometers wide that is illuminated through its glass support, creating an extremely small detection volume, just 20 zeptoliters. Nucleotides diffuse in and out of the ZMW in microseconds. When the polymerase encounters the correct nucleotide, it takes several milliseconds to incorporate it, during which time its fluorescent label is excited, emitting light that's captured by a sensitive detector. After incorporation, the label is clipped off and diffuses away. The whole process repeats, creating sequential bursts of light corresponding to the different nucleotides. These are recorded, thus building the DNA sequence. Our SMART sequencing harnesses the polymerase's natural ability to synthesize 10 or more bases per second over thousands of continuous incorporations, leading to high speed and long read sequencing. Additionally, our SMART technology design allows simultaneous multiplexing of thousands of ZMWs in parallel, all concurrently replicating DNA in real time. The data is assembled into whole genomes and analyzed by researchers. Our SMART sequencing technology will ultimately translate nature's ability to replicate an entire genome in under an hour to an instrument that will sequence a human genome in minutes for under $100. This unprecedented level of performance will completely redefine the field of genetic analysis and will enable new scientific advancements that will lead to improved healthcare worldwide. Pacific Biosciences SMART DNA Sequencing.